to the introductory lecture to protein prediction that has some focus on protein structure. Most of you will not have a background in biology. So let's begin with something that is philosophy. So somehow, you know, when you go back to the Greek times, Plato is the idea that you understand the world through ideas. That's physics, that is material sciences, and many of you may have chosen the subject you study, simply computer science mostly, because you like ideas, you like principles, you like to understand how things work in principle. There's the alternative explanation of the world, which historically started with Aristotle, and Aristotle's view is more on heuristics. Somehow it's the idea that you have a lot of boxes, you put a label on the box, and putting a label on a box means you understand, giving something a name. Biology deals with life. Life is a level of complexity that is beyond finding first principles usually. That's why biologists can only operate that way. That's why when you study biology, you begin to not be scared by names. Human, we have 20,000 proteins. Each of those has a name. There are many, many other organisms that are studied in the lab. And every one of them typically has a different name for the proteins. And there are biologists who know all these names. Okay? They're way in their capability of their brains. They're way beyond what I at some point studied physics. That is way beyond what I can imagine I can ever learn in my life. Now, introduction to biology for you, to some extent, means that I'm going to present names and names and names and names. One thing that if you studied biology would learn is not being scared means not everything, every name you really have to understand. It sometimes is enough if you map it to a class. So that name, this particular kinase is something that has to do with proteins. And my lecture will be full of terms that you don't necessarily really have to learn by heart. What I hope you carry away are two things. One, some feeling for the complexity of biology, in this particular example here for proteins, and one is some of the algorithms and the ideas that are needed to, in this particular part of the lecture, predict protein structure. This has several implications. One implication is you cannot go out there and find a book and read it, prepare that lecture, or have a companion for that lecture. It's almost impossible. Whoever comes now, you can explain to them what I said, or it is anyway taped. It means that most of what I say is in the lecture. When you need additional information, come and ask me, and go to the exercises. So the exercises this year, it's the first time that we do that, are designed very much to help you understand the lecture better, get additional material, get additional questions, work yourself into more detail, and these exercises create Wikipedia pages or wiki pages. And again, those wiki pages, I believe, I would hope, are a very good source for learning for the exam. The examen, let me repeat, Edda said that on Tuesday, the examen is entirely the score that you get at the end of the day. The exercises in that sense don't count. But don't count them out. The exercises will not be taped. So when you do not go to the exercise, there's no presence required. But when you do not go to the exercise, you clearly will miss things. In the past, we have, in fact, the final grade was a mixture between your performance in the exercises and the performance in the final examen. And everybody did not fare very well had we only graded by the examen. We hope that we will balance that this year by sending you to the exercises and making you prepare the examen. So we begin today with the introduction to protein structure. That is the list. The slides will be online, typically before the lecture in the future, and we will tape. I'll get back to that in a minute. This is shifting. This is just, essentially it gives you the dates. You can find it on the web, and while the lecture goes, somehow this will change. So, I don't have a photo of Carlo Di Domenico, who is recording the videos. Tim puts them together with Carlo or keeps them on the web. After the lecture, every video within a week, Jonas is no longer here. Jonas, typically within a week you manage, right? Yeah. So within a week or less than a week after the lecture, so in the past Jonas did that here, he was still sitting up there, the video lectures. So within a week you will have the video typically before the lecture you will have the slides on the website. That is shown here, and then further details for that particular lecture. Let me repeat also that the lecture will always start at 10, Tuesday and Thursday. There was a typo on Tom Online. The exercises from Edda, Andrea, Tatjana, and Jonas. Now, so this is an image of the expansion of the universe, and I take this expansion of the universe to talk about email. Email is one of those things that I personally find extremely dreadful, an extremely wrong invention. It was great 25 years ago, but now I can no longer handle it. Please, instead of doing rost@something.tum, send the email there. I will be here before the lecture, I will be here after the lecture. Come and ask me questions. If you really have to send an email, do it there. I discourage you from doing it, but you know, if you really have to. Carlo, yeah? Yeah? Ah, I thought you said hello. So you can send some email to him simply because he said hi. Carlo does the videos. And thanks, Carlo. Okay, so we begin today with genes, proteins. Some of the slides come from Marco Punta, some are from Andrea Schafans. We will talk a little bit about organisms, and some of that Edda did already. Edda presented the central dogma. I will briefly get back to it today, talk about amino acids, protein structures, a little bit about bonds, and I'm not entirely sure how far we get. And then we get to other components of 3D structures. Life, the players, what is common? So there's a tremendous diversity in life. What is common to life? All of these things, they don't have size in common. What do they have in common? Living creatures. DNA. So that is clearly one way of phrasing it. The basic thing is DNA, or the basic, if you think about it, we're sitting in a machine engineering building. If you want to build it, you begin with DNA. That clearly is what they have in common. There is a formal definition that you will find on Wikipedia. I'm not entirely sure whether I took this from Wikipedia, I guess I did. That means life. In fact, when I studied, there were 4 criteria. Now there are 3 added because we have to, we discovered new things that don't really fit to what we believe is life. So there's a couple of things that life is like, and that's typically biology. You can't give a clear definition. The major theory, if you want to call it that way, in biology is evolution. And evolution says, Darwin, the survival of the fittest. Now, who is the fittest? And you're immediately in a circle. By physics terms, when you begin the definition of a theory with something that is a circular definition, that is not a definition, right? Biology is exactly like that. And as much theory as saying survival of the fittest, more you will never get. It's a tremendously strong principle, by the way, and we will get back into that strength during this semester. These, in fact, again, is the way you reproduce, the way you grow, the way, in fact, you distinguish life from. One is on the operational principle, there's DNA, but again, you could actually build an artificial life with DNA. You could build a machine with DNA, and there are a couple of things that you need. Could you then still build artificial life? And can we build artificial cells? That is a different story. We can, in fact, do that. Are these then alive? Most likely they are. So if you put together something that looks exactly like a cell, then you create a cell. That is life. But still, there's something else here. Viruses, are they alive? They certainly have DNA. Some of them don't, some of them have only RNA. But let's not get into that little detail, yeah?
No, they are not, because they can't reproduce.
Is your background in biology?
No.
No? How do you know? From high school?
Yeah, from high school.
So viruses are not considered life because they cannot reproduce. Now comes biology. And you're totally right. This is what you will find in biology. That is what you find in most biochemistry 101 or cell biology courses that you learn as a biology major. That's typically what you would say. Now comes something that is called the mimivirus from Jean-Michel Claverie. This thing is huge. By definition, if you know anything about numbers, this has over 1,000 proteins. It can reproduce. It has a membrane. It has everything that makes it look like a cell. It's still defined as a virus, and there are biochemical reasons why it's still defined as a virus. But what I want to say with this here is biology lives with these labels, and then suddenly somebody discovers something. So this is just a few years old here. That is mind-boggling. We had no idea that something like that existed. Let me give you a background. Typically viruses, so your flu viruses, they have 5 genes. This has 1,000. Sometimes people saw big viruses, 15, 20 genes. 1,000 is amazing. Ebola, for instance, has just 10, or less than 10, in fact, 9. And that reproduces. So is that now really life? The question is out there and we cannot answer it. Most biologists will still teach your answer. Viruses cannot reproduce, since therefore they are not life. But biology is more complex, or life is more complex, is what I want to say here. And in fact, this mimivirus is not an exception here. There are even bigger ones, and many different types of those. At the days of Leonardo da Vinci, the idea really was, and that Leonardo da Vinci is very much a Platonist in some sense. He signs up to this idea that you understand the universe from first principles. This idea of the human put into shapes means that these simple shapes, triangles, circles, they in fact reflect our understanding of the universe. And man is part of the universe. Sorry. And by painting him in these circles, the idea was you have some understanding, and you show somehow how this whole thing fits into our understanding of simple principles under which the universe is organized and under which we also live. There's still this idea somehow around. The central dogma. One way of phrasing it is there's DNA, there's RNA, there are proteins. Proteins are the machinery of life. DNA in some sense is the information. You can also phrase it, and that's the slide from Edda, you can phrase it in terms of information flow. So the information flows from DNA to RNA to protein. This circle is that somehow the way DNA is processed is also written in the DNA. So the information, how you process the gene, is written just before the gene, or in the gene, depending on how you define that, but in the DNA. And then there are all kinds of circles here. Sorry. This is in fact the DNA, the double-stranded helix, that is translated into an intermediate strap, into the RNA, and then becomes ultimately the protein. The sequences of proteins these days are done through something that is called next-generation sequencing. Next-generation sequencing started about 10 years ago, and every year there's a new generation of next-generation sequencing. So today that typically is referred to as deep sequencing, because people no longer really, the speed at which this has evolved is mind-boggling. In fact, by far outpaces the development speed of computers. This machine here fits nicely. It's less than a printer size, fits nicely on your desktop, and it sequences one of us on a typical day. Now this machine, the price for that is in the ballpark of a little bit above €1,000. If you read out all the details about the raw reading of this machine, then this machine would produce 5 terabytes every day. And you immediately see that we have a problem here. You operate this machine a year long, and you know this is just your desktop. Sequencing everybody here in the room or larger groups will take much more than that and produce an immense amount of data. How do we actually move these data around? What do you believe? How large sequencing centers that sequence thousands of individuals in a very short window of time. And now they want to move that to some other place, say from Boston, where the Broad Institute produces a lot of data, to, for instance, Leipzig, where they work on ancient people, ancient human. How do you believe this has moved the data in 2015?
By plane?
Yes, exactly. This is horrible, FedEx. So you put it on a disk, on a hard disk, and you fly it. That's exactly the way you do it. 2015. At the immense speed of internet, we produce data, they produce data that cannot be sent through the internet. It's just too much. Anyway, there is this idea, no, there's not the idea. DNA is nucleotides, it's a 4-letter alphabet. Proteins are amino acids, it's a 20-letter alphabet. The way you go from 4 letters to 20 letters is simply by using so-called codons. Codons, you take 3 at a time. So 3 nucleotides together code for one particular protein, amino acid. And that is shown here, the codon G with a C with an A. GCA codes for an A. And that is an amino acid here. This happens to be called alanine. The 20 amino acids, the 20 natural amino acids are shown here. 20 amino acids, essentially, and again there are minor deviations to the statement here, but for the course of this lecture, it is true that life only uses 20 amino acids. That is not because there are not other amino acids around. We could have others, and in fact, again, so there are exceptions, we have some others, but life didn't use them. Why? In fact, there could be 200. Depends a little bit on how you do the chemistry. You could easily, I don't know what the natural number of what you would call amino acids is. Again, in life, 20 are used, and that typically is referred to as amino acids, but you could create others. Why didn't we use the others?
Maybe it's enough.
Why is 18 not enough? That is a great statement. So is your background in physics or something?
No, biology.
You're a biologist?
Bioinformatics.
Oh, but then you're sitting in the wrong lecture, you know that. Oh, seriously. You should be sitting in a different building in 2 hours from now, at 12:30.
Okay.
Yes, that lecture is not for you.
Okay.
It's up to you, but this is absolutely not designed for you. If your background is in bioinformatics, go to, and if you can afford it in terms of time, so the bioinformatics lecture is at 12:30. Maybe we will move that, but we will decide that today. In the informatics building in what is called Finger 9, I don't know whether you understand what that means, on the first level in the seminar room, which is the second to the right when you go through the glass door. So maybe 20 is enough. And that is somehow, I asked you whether you're a physicist, because in physics, somehow the background always is you try to explain with a minimal number of assumptions. And again, we have somebody here, Occam's razor. Some of you may have heard of Occam. Occam has lived in Munich, by the way. So Occam's razor is the attempt to cut away anything that really is not needed to explain something. Explain your observation with the fewest number of free parameters is the way you can formulate that in terms of statistics or machine learning. Now, that could be true, what you say. However, we cannot really explain why it's not 18 or why it's not 22. We can do things with amino acids that we have not seen before. They are used for protein engineering. Protein engineering means you take known proteins and try to do something new with them. And often you put these new proteins into organisms and try to make them do something new. Like make bacteria eat plastic or something like that. That does actually work. Or digest drugs in the water. There was just one such example that was done a year ago by students in Weinstefan in the context of a biochemistry class. And in this particular case, again, we did not need additional amino acids, but there are examples where you can actually do something new with an additional amino acid. In some sense, you could then argue, therefore, the principle that you don't need more is valid because you can do something else. We can do many things that proteins do by removing one amino acid. So in that sense, in the other direction, your statement would not be right. But the truth is, whatever you suspect, whatever anybody suspects why it's 20, we don't know. As simple as that. We actually, to be more clear about it, have no clue. We have many explanations. You begin in one direction, and we can improve over what you just said in a moment. There are people working on that. But all of these are explanations that are not predictive. In the sense, they typically do not start from 0 and then say it's this number, and then I observe it's exactly that number. Instead, they observe something and they rationalize why that what I observe could be right. This is what you do very often in biology. And that is a tricky bit. Now again, in my eyes, I don't believe we really know it. There is this idea around that, in fact, life started as RNA. In terms of the code, genetic code, I explained, or in terms of the dogma, from DNA to RNA to proteins, I said RNA is an intermediate step. Still, historically we believe that life started with RNA. And RNA cannot do the type of enzymatic reaction, cannot easily do the type of enzymatic reaction that it may have to do in order to become life and replicate itself. Because replication, as you correctly observed, is a very important example or aspect of life. Since RNA cannot completely live alone, it needs to find something that helps it to live. And maybe it had this great idea to hijack amino acids, build little peptides, and putting these peptides together, it could live. And maybe this is exactly how life started. And maybe what we see as amino acids is biased by RNA wanting to start life in some sense. That is one idea out there. And Andrei Lupas at Tübingen is one of the big pushers of this idea and has a lot of evidence that is very suggestive. But that was just a side story. Codon. We have 3 nucleotides that together form the 20 amino acids. A 3-mer, you combine these 4 different. Now, 4 by the power of 3, as you all know, is 64. So it's much higher than 20. And already you see here, in terms of those are the 20 amino acids, and you already see that some map to more codons than others. So this M here, there's only one way you can get to an M. It's the ATG. And others, you have many ways in which you can get to them, right? Why is that? It's another one of those questions where we in fact do not quite know the answer. Again, it's another one of those questions where we have many answers, but we don't quite really understand it. That is reality. So one thing that you also understand, however, you could not do with 2. So 4 by the power of 2 is only 16, so you could not do it with 2, right? You need 3. Or you would have to throw away 4 amino acids. Actually, there's another one. You may have seen that here is this red flag saying stop. Stop means this set of codons. So if you encounter those 3, then you will stop translating the DNA to RNA to protein. Stop codons is what they are called. So that is in fact another piece of information that will not end up as an amino acid, but it will decide how proteins look in some sense. Okay, so say you only needed 1 stop codon, then you could only live with 15 amino acids, and maybe this is exactly your argument. Maybe 15 is just not enough. Maybe that's why we need 3 codons. Biology is full of speculation. Let's just see translation in action. And now that is not very impressive.
Small red tips attached to the transfer molecules.
Okay, let's go back.
There are different transfer molecules.
Sorry.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So the red thing that you saw going off there to the right top, that was the protein that was being formed. Now, the ribosome is a machinery. I said all action is done by proteins. The ribosome is a machine that contains about 100 proteins, so it's a large complex. And in fact, a lot of the processes in the cell are done by these large aggregates. Ribosome is just one example for that. Is there a question to this movie? Okay, let's get back, to entire organisms. I said we have about 20,000 proteins, and we'll get back into that number in a minute. What do you believe the smallest living being, so viruses are not it, bacteria, what do you believe the smallest bacterium has? How many proteins? We have 20,000. What's the smallest thing? Any idea? So you're not counting for this one. Actually, do you know it? No. But even if you study biology in some sense, you still may not know. So the smallest thing, in fact, is, well, one of the smallest things I get in a minute is not Haemophilus influenza. Haemophilus influenza was, in fact, the first entire organism that was sequenced. With 40 papers, and that has about 1,500. Mycoplasma is the smallest, and that's in the ballpark the smallest organism, and that has in the ballpark of 600-700. Yeast, and let's look at the dates here, that's 95. Yeast, about 19 years ago, the first eukaryotic cell was sequenced, that was yeast, and that had taken many years to be done. The first multicellular organism, so yeast is a single cellular organism, eukaryote. C. elegans, the nematode, a few hundred cells, a few hundred neurons, shown here, sequenced in 93. Drosophila, insect, here the numbers, 500, 1700, and so forth. We go to the first eukaryotic, 6,600, Drosophila, worm. And now we get into something that in fact is another example of biology. So the estimate from 1999 for human looking at that in some sense was not surprisingly 100,000. So it was assumed that in fact we have 100,000 proteins. Now the remarkable reality here is the human genome sequencing was done in 2000. It was supposed to be ready by January 1st, 2000. Did not quite work out. The reason why it didn't quite work out, the major reason why it didn't quite work out, is that there was a press conference in one of the large centers that did the sequencing, the Sanger Center, the EBI at Hingston in Cambridge, England. And at that press conference was in October 1999. October 1999 is after about 8 years of sequencing. Millions of dollars, 8 years of sequencing in the human genome. In October 99, there was an announcement. We were a little bit off with the number. It's not quite 100,000. It's 30,000. This is after 8 years. 2 months before you want to publish and go to the big press. You suddenly say it's not 100,000, it's 30,000. So whatever your background is, for biology, that's a minor difference between whether you have 100 euro in your pocket or 30 euro in your pocket, right? The speed of light, 500 years ago, the estimate of the speed of light was closer than 5% to what we believe today, okay? Biology works 8 years on a large project, and this is a factor of 3 of. And this happens in biology again and again. And that is not because biologists are not clever. That's just because it's immensely complicated. And in fact, that was the reason why the publication was not on January 1st. They couldn't believe it. And they had to reassemble. Reassemble means, so the way sequencing is done is what is called shotgun. So you get small pieces. When you have these small pieces, in the end, you have to map them to where they fit in the genome. And you cannot do that before you have all the small pieces, right? You can map some of them, but they may go somewhere else, but you need the whole picture. That whole picture they only had in summer in 1999. And then essentially they redid the calculation 5 times before they could admit that they were so wrong. Again, biology is immensely complicated. And interesting, that is exactly why, so today the number has gone down a little bit. In 2000 we believe the number was 30,000, now we believe it's more in the ballpark of 20-something thousand. So the current estimate, in fact, even that one here is outdated, as you see, 5 years ago. Today we really believe it's more in the ballpark of 20,000. Now, why is it that we move downward here? Is because some people are not very comfortable being in a ballpark of number of genes that is comparable to this worm. C. elegans is this little thing that you cannot see with your blind eye. I need a magnifying glass to see it. And it essentially eats everything. Every piece of DNA you throw at that thing, it eats. And it's a darn dumb thing, right? It has as many proteins as I. And some people are not happy with this realization. And that, I believe, more than anything else, was the reason why we kept estimating it higher. The technical reasons, really. It is not that easy to get from a genome to that estimate of genes because we still don't quite understand that. Anyway, now we have this entire genome. Do we now understand how life works, how a human works? And Anna Tramontano, I unfortunately don't have the picture here, I don't know why. Someone didn't make it. Anna Tramontano says, like for every good manual, so imagine this entire genome now is like a manual. And for every good manual, you hardly ever find what you're looking for. And if you find what you're looking for, you don't understand it. At least this is my experience with manuals, my frequent experience with manuals. That's one way of looking at it. Another way of looking at it is, imagine I gave you this, but I wouldn't give you the map. I gave you the parts. You'd never seen a car. Okay, having seen it, in this sense we have seen human, but we don't, by no means, from the genome, putting it from the genome together, this is not like you have seen a car, or having seen a car helps you more to put these pieces together than the genome of a human. And actually we're not talking about a car, this is much bigger. This is an A380 or something like that. And put that together from the parts without a map. And that is a little bit the situation we have. We have pieces, and we still don't quite know what they mean. And the last 15 years were occupied with trying to understand how they fit together. And in trying to piece it together, we learned more about the complexity. One important aspect, again, is diversity. When the first human genome was sequenced in 2000, the idea was that essentially one is enough. There were two efforts, you may have heard that. One was I briefly alluded to that in this picture here, with Bill Clinton and Craig Venter and Francis Collins. So Craig Venter represented the company, Celera. That was one human genome. And Francis Collins represented NIH. He is now the director of NIH, has been the director of NIH over the last 5 years, but he was the leader of the public effort that included many countries. And they did theirs. Celera sequenced one person, and you can easily guess who that person is. The public effort, of course, couldn't do that. They could not sequence Francis Collins. Anyway, Francis is not the type of person who would ever have done that like that, anything like that. Instead, like any good public thing, it was sort of a democracy kind of thing, 5 people. Who they were was not revealed. But still, in 2000, we believed that wouldn't matter. We believed that essentially there's one human genome, and the way we differ, we map as differential to that reference genome. We have this one thing, we share it all, and we deviate from that one thing that we share all in some places. We have one. When I sequence the next person, I just describe that next person in terms of slight differences. Today we know this is absolutely not true. In fact, there are, not sure there's an African in the room, but for Africans, the sequencing has to be redone. In fact, the entire way of the way sequencing is done is currently not working for Africans. There are major mistakes that happen. We believe, many people believe, and clearly I believe, that human came out of Africa. So therefore, getting to that point of sequencing Africans is absolutely essential because those are our roots. But this is 15 years later. We know that the story is more complex than we thought. Many things have changed over those 15 years. Back again, the dogma is still the same. We DNA to RNA to ribosome to proteins. Proteins have a structure. Now what does that mean? You take this stretched out amino acid chain. They differ in the number of, imagine amino acids like pearls. You string them up. And that is exactly in this movie, you saw that. When they come off the ribosome, they are a string of pearl chains. The only difference, there are two differences between two different proteins. One is they have a different number of pearls. And two, they use the color differently. So they have 20 different colors. Imagine this, the 20 amino acids are 20 different colored pearls. And the way you string them up differs. Right? You use different sets of colors and different compositions. Now, when I take one particular pearl chain, and I throw it in a solution, it adopts a unique three-dimensional structure. Unique means that I do that again, it does exactly the same structure. Say the shape is like this. Throw it in again, it will be the same shape. This is the assumption. For some proteins that is the case. For other proteins it's slightly more complicated. But for the course of the lecture, this assumption will hold. Essentially this is true. Now, when the three-dimensional structure is formed, the details of that three-dimensional structure lead to how it functions. You can imagine this really like key and a hole. You have a certain key that fits into a certain hole. This is the principle in biology, in proteins. You really, in principle, can see when you look at the proteins in 3D, and we will talk about how you can do that. When you look at them in 3D, you have somehow can guess, if you're an expert, what they do. Key fits to a hole. You can say, well, looking at the key in the hole, you could say, this is the hole it fits, and this is the way it doesn't fit. And that really works for that in proteins. And now you immediately also see why the particular pearl chain always has to adopt the same three-dimensional structure. If not, it would not do the same function. If one particular protein would not do its function, the story would get complicated. In fact, we have examples like that, typically through mutations or other damages, and typically they lead to some kind of problem, disease, in many cases at least. So again, DNA to RNA to protein, that's an intermediate step. Just another example here, one paper that was now 10 years ago showed that this intermediate step is not an intermediate step. that there's a huge universe of RNA, what I call an intermediate step, that has an own life. And this is a whole universe that I suppress. Again, biology is more complicated. We still, essentially the dogma holds, and for the course of this lecture, proteins, the RNAs are intermediate. The particular amino acid sequence forms a unique three-dimensional structure, and that is unique for the function. Let's call that the dogma under which I will operate for the rest of the lecture. And mostly that's true. Now, can we compute life? It's a very, very tough challenge, but the problem is we still don't understand really the book, the genome, and we don't even understand the principle under which they are formed. Nevertheless, and here's Craig Venter again, you may remember that I just said in Celera he was one of the first people who was ever sequenced. He was also the first to synthesize an entire organism, and was a type of Mycoplasma genitalium, which I said was the smallest, about 500 proteins large. And that was an entirely synthesized artificial thing. The first generation did not reproduce, so by that criterion it would not be considered life, but they refined that. There's Mycoplasma now that essentially comes out of chemistry. Now, is that artificial or not is a matter of debate. Because in the beginning here, they simply copied what they had before and removed things that they really believed they wouldn't really need. Large field that has come up since. And that is done before we really understand. We cannot compute life, but we can build artificial life. I believe understanding and computing life would be great. And I believe it would help you, if we understood more, will help you over the next 10 years to get a more specific diagnosis for any problem you may have, health problem you may have, and may also help you to lead a better life, personalized health. And I will get back to that over the course of my lecture. What are proteins? So what they do is they defend against antibodies, so intruders, viruses and the like. They form structures, hair and collagens. They do enzymatic activities. They do communication, ligand binding, storage, and. Everything in life is done by proteins. This is just one image of different protein structures. So the ribosome shown here, they are not to scale. So then the relative sizes here are not right. Hemoglobin is the one in the movie that you saw, came out of this, and you immediately see the movie was more to scale than this one here. There you saw that this is much larger compared to that. Ultimately what this picture here shows is there is diversity of length, of shapes, and they create a diversity of function. And then there is more here, that's a virus, and collagen here, actin fibers. Actin fibers, you can imagine, are in fact here, muscles are the tracks in cells. A lot of the signal, a lot of the transport of material is done through actin fibers or myosin. So you can really imagine that there is an ATP molecule that moves, is an active train system. So the train moves by itself. It moves on tracks. It's amazing. I'll try to find a movie for that. That is just a protein, or an aggregation of protein. There's one unit that is replicated here. It's a lot of the same thing that forms this long track. That's how it looks in terms of sequence. 20 amino acids strung up in a simple word, if you want. This particular one here is the potassium channel is ATPase, not the potassium channel. And the potassium channel, I can't remember how long that is. This is subunit A, I don't know. I guess this is about 400 residues long, 400 of these letters long. Some facts about proteins. How many are in human? I already said that. 20, we currently believe it's about 20,000. How long are they? Anybody? Everybody's included? People with high school background or bioinformatics background? How long are proteins? So I showed you one before, and I said that is roughly 400 letters. What I did not tell you is whether there's an average. I showed you on the slide before, on the gallery of proteins, I showed you sizes. I showed you actin, but I told you those actin is in fact many actin molecules strung together. A single one is what I showed you before. How long are they? How many amino acids do they have? What do you believe? Anybody?
Several thousand?
Several thousand. Well, so that already would be an interesting point. No, it was said subunit. Okay, point taken. Several thousand. Anybody believes they are shorter, longer? No, you guys don't count. They don't count because they have heard at least that part of the lecture before. So they are 35, that's about the shortest, to 30,000. Several thousand is totally right. Completely in this spectrum here. The median is around 400, but median really is a misleading term. The important thing is that there is immense diversity. Do they consist of units? And that already somehow I said, they have regions, they have domains, they have subunits. Most of them, most proteins that exist have more than one. How many proteins do you believe are known? We have 20,000, but we clearly know more than human. So this lecture will be about predicting protein structure. It will be about, ultimately you will find out that it always starts in biology by seeing, observing a new sequence and comparing it to the knowledge that I already have. Knowledge I already have, it begins always with a database of protein sequences. Every new discovery will be compared against that, and that essentially is exactly the type of algorithms we use in computational biology. And the question really is, can I map this new observation to something that I already know? Through the strength of evolution, that is the strongest signal that I have, the most meaningful thing. So how many proteins do we know? What do you believe? Must be more than 20,000. Anybody? Billion. Anybody believes there's more than a million? A relatively tiny minority believes it's, I'm not entirely sure. Who believes it's a million or less? So that group is clearly larger. Who believes it's more than a million? Yeah, this group is sizable by now, but it's smaller. So at this point, the number is over 55 million. And that is not the complete set that we know. That is the way it grew. So the growth of the biodata is way more than a factor of 2 over 15 months. Now, what makes this, I'm putting the 15 over 2, way more than 2 over, so the computer growth is and I will at some point map that. Computers double every 18 months. That was a statement essentially true until 1999. Since it has slowed down significantly, this is more. But what makes it even worse, in some sense, is that when we begin with computational biology, we always compare things. We look at everything that comes in new and compare it to what we know before. So actually we square this problem, because the comparison is always a square operation. I use it 1 against everything. And the more I get, the more against everything I have, and the more I outgrow the computers. And that is beginning to be a real problem in the field. Or challenge, or the point where exactly computer science is needed. Depends on how you look at it. Now, in comparison to this one here, so this is how the sequences grow. This here is how the number of proteins grow, for which we have some idea about what they actually do, for which we have an experimental biologist who did some experiment. That's in the ballpark of maybe 400,000 at this point. 55 million sequences we know. For roughly 400,000, we know something about what they do. Okay? Okay. So it's a fraction. Proteins are gene products, are the machinery of life. done through these codons. Let's skip this and let's get into there's something else I forgot. It's obviously too late today. The lecture will be 90 minutes. Should the lecture contain a bio break? No. Good. Oh, wait a minute. Actually, we need really, I heard 3 no's, but I was a little bit too fast. Can I really have a show of hands? Who wants a bio break? 3, 4, 5. 6. Who doesn't? 7, 8. Sorry guys for the bio break. You're highly welcome to leave, but I will not stop. So let's begin with one thing. When we look at different types of organisms, for instance bacteria or eukaryotes, I said proteins use a different color scheme. So they have all the 20 amino acids, but they combine them in a different way. What do you believe if I looked at the average over all eukaryotes or all prokaryotes? Would they differ in the color scheme, in the usage of one of these 20 amino acids? Well, let's just give you the answer. How can you know that? Most biologists don't know that. So those are the 20 letters. Those are the 3 major kingdoms of life. Eukaryotes, that's where we sit. Prokaryotes, the bacteria and archaea bacteria. This should read bacteria. Those together are prokaryotes. Now, the height of the letter is proportional to the usage of that in that kingdom. Again, those are the 20, and essentially they are the same. Right? Nevertheless, organisms do differ in their codon usage. That does not quite mean that they differ in their amino acid usage, but they also do. But over the course of the kingdom, that averages out. There's a slight difference in the amino acid composition, but there's a big difference in the codon usage. Mind you, codon is the 3 letters that I put in order to translate it to an amino acid. The amino acid essentially is the same. The codon is different. And again, you see another reason, another possible reason why, and there are reasons for that. And another possible reasons why you need some amino acids to be coded for by more than one triplet. Okay? Reality and images. A lot. What I'm going to present over the course of this lecture are what we perceive as the truth. Protein structure. I can measure, not easily, and we get back to that, I can measure in a particular protein structure every position of an atom. That's a reality, right? That's an image. It's something that is absolutely real. And that is not quite true. We create images, and they suggest a reality, but it is not reality. Let me give you a few examples. Let's begin here. What do you see? Houses. Georges Braque. Houses. Yes. Good. Where's that? I still didn't get, what did you say? What is it? Did you say what it is? I didn't hear, no. Anybody? So I can give you one hint. It's not houses this time.
Flowers.
Flowers?
Yeah, background with branches.
Yes. That's true. That makes a lot of sense what you say. And you want to lie down in it with the sun, right? That's Mycoplasma genitalium. That is in fact the smallest creature, or the smallest bacterium. And here are in fact the explanations what that means. That's the cut through a eukaryotic cell. What's that? It's blue and red, it looks nice. That's not my question. What is it? You have to be a little bit louder. Yeah, great idea. Wow, you have a sense for molecules. So you, first of all, you're absolutely right. This is molecules, and it's a biomolecule. It's not the DNA double helix. This is a protein. Now, and that is what I mean. So those are two different representations of exactly the same thing. I did an image of all the atoms in that protein. Here, I simplify them, and I sort of look at the major, what is called C-alpha traces. So the major part for every letter. Every letter here is represented by one ball. And it could be a DNA double helix. The moment I don't use this representation of a ball for a letter, but I use a representation that sort of sees units. The rods here, so-called alpha helices. Alpha helices are, I get back to that in a minute, are defined like that. You immediately see that no longer is a DNA double helix. It's the same thing. It's just visually, I highlight, I group together differently. And you see something else. Now you may have said this is some metal or something, but you would most likely not have answered again that this is a DNA double helix. First of all, if you knew it's a molecule, you wouldn't, it does not look like a DNA. And on top, it does not look like a molecule at all if it could be a machine, right? Here is another molecule that I call dehydrogenase. This, in fact, is in the ADH, so-called ADH5. This is important because some of us in the room have that and others don't. And this is very important if you are stupid enough to want to drink too much alcohol. And if you don't have that enzyme, then this is going to be a worse decision for you than when you have that enzyme. So some in the room here don't have that enzyme, and for them drinking a lot of alcohol is even worse an idea than for those who have that enzyme. That enzyme is digesting alcohol. And that, in fact, what you see, or what you may see here, is these things, and I'll get back to that again, are helices. There's something else here, I'll get back to that in a minute. But you actually have a twofold symmetry that you may see along this axis here. On that side and on that side, you have a duplication of the same molecule. What's that? Not houses, not DNA, not protein. What is it? Now I'm going to listen. So let's just write this down. It's very clear that nobody who has never seen that before knows what it is, okay? So just let your imagination run. Spiders. What? Bias? Okay, viruses. Can I have? I want to hear a couple of things. Just be louder. Just say something louder. Yeah.
Burning paper.
Paper.
A burning paper.
Burning paper. Fire. That I have heard before, but that's funny because several people on this have said fire. Burning paper I've never heard, but yeah, I believe that's what you mean. Fire. Yeah.
Tortilla chips.
So soon is the lunch, right? We're hungry. Maybe we should have an eating break. Tortilla chips. That's an interesting one. Yes, that's true actually. I see what you say. Okay, anything else? I can immediately tell you, so far nobody has done it, but we have something that is close to it, yeah? A manifold? Well, that is a little bit descriptive, right? You have a manifold of objects that you put together. But what does it mean?
It's like in a hyperspace maybe?
Yeah, but again, so you're too descriptive. You have a too mathematical or whatever that mind is, or engineering mind. You take it apart and you say this object has been drawn by a manifold of shapes, right? But that doesn't mean what it is. That's true. That's the way it's done. That's not what it is. What is it? Okay. Okay. That's what it is. It explains how a soccer player is dynamic. And in fact, Boccioni has done that using this photo. So this is a direct rendering of the same thing. in some sense. And clearly it's a manifold of impressions. Clearly the idea of fire has something, because that is this idea that there is a really dynamic fire energy. But again, what I want to say here is this is one reality. When you see that person move, that's another reality. When you get this one, you see another aspect of it. In fact, I would argue that at least some in the room saw more energy in this than in this. This looks more frozen. This would not suggest fire to you. If you saw this guy move in this defense situation, you would see that there is actually fire in it. But you would rather see that in this than in this. Right? Again, images and reality. And this comes actually from Marco Punta. The same is true here for our proteins. These are two renderings of exactly the same molecules. Here and here. It's a slightly different grouping. It's a slightly different visualization. It's not as extreme as this difference here, but it is. So what is reality? What I want to say, and I'm going to bring some examples here of amino acids. I'm going to bring some examples of energy, landscapes. All of these are explanations that are like images. We present reality. We don't capture the real thing. The particular position of the atom is not the real thing. Just like you said manifold to this painting, this is an observation, this is taking it apart, this is the constituents in some sense, but it doesn't explain the object. Right? And the same thing I'm going to do now. I'm going to give you the constituents, but this is not going to explain how a protein structure does its function. Okay? But the lecture is about understanding the constituents and understanding what we can do with computational tools to map what we observe into what we know. Amino acids. Amino acids essentially have two different, can be perceived, presented, imagined to contain two different components. The so-called side chain and the so-called backbone. Backbone is typically the thing that all 20 amino acids have. Side chain is the thing in which they differ. If you go along with this image of a pearl chain, this is the ball, this is the color in that image. Again, I'm very misleading here. Now, as an isolated amino acid, then you string them up by forming a dipeptide bond. This dipeptide bond has to do with electron sharing, and in fact, you push against a force. The moment you have done that, you gain energy. So you go over some, in order to do that, you go against a wall and then you gain. That's why the dipeptide bond is so strong. Again, the way you visualize a dipeptide bond is essentially, again, what you would have here is the backbone, the two backbones that you join, and here the bond as some long rod, which is an abstraction of the fact that this electron that you share is observed on different sides, right? Or in a circle around it. You could also imagine it like this. And both these images here transfer some aspects of that bond. None of them is more right than the other. So we don't understand how proteins work, but actually this is on the level of two atoms. And some models that we have are better explained having this in your mind, and some are better explained having this in your mind. Or in this one, here's the electron surface in some sense. The sphere in which the electron can be found. Different images. Anyway, so now I string up two, I form dipeptide, I string up more, and I begin to form the protein. Again, I said that the observed proteins have 35, so you would, instead of, in this particular case, that would correspond to 4 amino acids. So you would string up 35 of those, and you would essentially have the shortest protein. And again, one representation is like this, the other one is like that, where in fact the backbone here and the side chain. The side chain is in fact, in this particular case, is the other way around. I mix the colors. No, I didn't. So the side chain is typically, for most residues, is larger than the back bond. And now here you string them together, you see them strung together, 3 amino acids, read the side chains that stick out from the main chain. The way the peptides are formed, they form a plane. The backbone forms a plane that restricts the way they can fold, adopt a three-dimensional structure. I call it rationalization of biophysical features of constituents. Many people believe what I'm going to give you are explanations why proteins work the way they work, are explanations of the energy levels that are involved, and those in fact explain how proteins fold, how proteins exist. I'm calling it rationalization because we don't understand the physics well enough to predict a protein structure. I said that you take this pearl chain, you throw it into water, it adopts a unique three-dimensional structure. If that is the case, if I can take 35 amino acids, put them into solvent, and fold them up, that means that the entire physics of the problem is contained in these few constraints here. Water and 35 residues. So, if I understand the physics well enough, we are talking about atoms. It has nothing to do with the complexity of life. Then I should be able to predict the 3D structure, right? There's nothing else that comes into my game. Do you believe I can do that? You all know a two-body problem, Earth and Sun, Earth and Moon. We can easily compute this system. We could do that essentially here for atoms too. A three-body problem, water, H2O, or Earth, Moon, and Sun. You already know this is very, very complicated. Today's computers, we can do that. But we can no longer do that analytically. In the protein of 35 residues or 40 residues, we have a 40-body problem. Cannot do that. There's no computer in the world that can fold up anything of the length of 30 residues. That's not quite true. Proteins fold on the order of a minute. No computer in the world can reach this within for 40 residues. The largest computer is Anton 2 now. It stands in New York. It's a special purpose machine just for, in fact, folding a protein. That can do some millisecond to almost second of it. And that's the biggest thing we can do. So we can do some of that, but the whole thing we still can't do. And we cannot, certainly not do that for things much longer. And that indicates that, in fact, we don't quite understand the physics behind it, or biophysics behind it, but we rationalize them. So the first thing begins with the fact that side chains here have differences. They have different biophysical features. So one feature is some side chains have charges. Positive charges, the R and the K. Negative charges, the D and the E. Some of them are polar, so they separate essentially out the charges. Some are hydrophobic, so they want to aggregate, string together. There's double bond there. I'm not sure how much detail I want to present on bonds and for folding proteins. And I'm going to go quickly over those slides. What is important here for you to remember is that there are different types of bonds, and that there are different types of side chains, and that different types side chains have different features. Charges, polar, hydrophobicity. Let's just stick there. There's a little bit more detail that will come in the exercises. There are ionic interactions that come, of course, from charges, and then there are salt bridges, again, charge-based, pi-pi. One very, very important thing is hydrogen bonds. Hydrogen bonds, H2O, ionic interactions, salt bridges, so Ke, so negative, positive, metal complexes are cations, and there are examples for that. Now let's get back to something that is more important, more crucial. When we when I showed you structures, I always showed macroscopic things, macroscopic substructures, things that you could immediately see on the screen, although you had not often seen structures. And they are often referred to as secondary structure. It's this sort of second thing, or whatever the rationale for the word is. And they're here. The first one, primary structure, secondary structure, tertiary structure. Sometimes people come and talk about the quaternary structure. A quaternary structure essentially means how two proteins are binding together, or how they form. Remember the ribosome, I told you 100 proteins, so somehow that is an aspect of a quaternary structure. Now, back to the two secondary structures. One, in fact, is the alpha helix. So, when you form the dipeptide bond that I showed you before, when you group together two amino acids, you can only rotate around an angle. So my fists essentially are the side chains now. And there is a certain way I can rotate and not in another way. There is a slight rotation that comes with the dipeptide bond. That slight rotation induces sometimes a twist. And you begin to see that here in this thing. Oh, that's the other one. Oh, wait a minute. The slight twist that you saw before, a helix, right? Here's another thing that you have seen before. So that is one chain running. Maybe it comes back here and is running again. Maybe it comes back here, is running again. So you have essentially like fingers. And certainly on the alcohol ADH5 and the alcohol dehydrogenase, I showed you an example of two arrows. There, I believe it was displayed as arrows. And that essentially is one part of the protein going back here, and there's bonds in between. And somehow they are stabilized like, fingers when you swim. That again is the protein structure. The moment in that protein structure is, now I show you every single backbone as one ball here. And here I simply take the balls out and you begin to see secondary structures. You begin to see here this sort of helix kind of thing. Here the actual helix. So here's much longer. Here's just one winding of something that could be helix. You may see that there's something here that looks like a helix. There's one wind of a helix here. And with a little bit of imagination, you may see that between these two here, you have something that I showed you before. These parallel things. Where two lines run next to each other and could be stabilized through binding here. And indeed, in the coloring of regular secondary structure, red is helix, and that is so-called strand. Here, the helix. The helix is stabilized by hydrogen bond formation from one residue here to I+4. So 1, 2, 3, 4 residues bind through a hydrogen bond, and they stabilize in every one of them. So this one goes here, this one goes there, and so forth. So you see this array of secondary structure that stabilizes the helix in that direction. The beta strand in yellow is stabilized by hydrogen bond formation between these two strands. Okay. Now, Ramachandran, is a molecular biologist who sort of first had this idea of mapping structures to the backbone angle here. So asking, when I look at the variety of all the protein structures, what type of angles do I see? Right? You can describe a protein structure by its coordinates. For every single side chain or backbone, or for every single atom, you can define a 3D coordinate. Or you can define it by these angles. Right? What do you believe you will see? Will you see everything? Well, already if you think about the problem, so you string up things, and I said that you can rotate somehow, but you cannot totally rotate. So clearly, the map will not be completely full, right? You will see structure in this space. It may have been a little bit surprising how much structure you see. Most of it is, in fact, is totally white, not used. And most of the observations are in this area. And this area, this is the alpha helical area, helix and beta strand. This is a little bit more the density, the actual density, but still you see that mostly white. Now, back again, I said those are strands. Back to the reality of many, many words. Here it's called sheets. Just a sec. The reason why it's called sheets here and why I talked about strands, one thing is a strand, you put them together, you form a sheet. That's what this one is called, yes?
When you talk about these binding together, is it always that the primary bond is between backbones only?
No. So for a secondary structure, so the question is, when I talk about these and these, those are secondary structure. They are always between backbone, they're essentially stabilized by backbone hydrogen bonds. Now the question was, when I look at all the interactions in a protein, are they always backbone hydrogen interactions? Absolutely not. So I should have spent a little bit more time maybe on the biophysics before, but the ionic interactions are between side chains. So side chains are very essential, not for the formation of secondary structure. They're essential for everything on top. Let's call it that way. So they are not essential for the local structure, you would argue, but also that is not true. Because you can immediately see, or you could imagine, you cannot see that, but you can imagine that sometimes a reaction, so say you had, in fact, an ionic interaction that may bring this piece next to that piece when it folds, just because there's an ionic reaction. The hydrogen bond is weak. So the driver of the process would then be the ionic interaction, and then the lock in place would be the backbone interaction. So you would have two different aspects of the energy. One driving the folding is the process under which a protein goes from stretched out chain to a unique three-dimensional structure. So some drive the folding, some lock the folding in place. Secondary structure is locked in place by backbone hydrogen interactions. Now, here again the different angles. Those are simply different representations of the same thing. Here you see the chain as it goes. Here is a rod. Which one is more real? And again, you may argue this is a simplification just for visual guidance. But it's more than that. Because remember there are these hydrogen bonding network. So if you think about this as excess, as something that would intrude into this part here, into this groove, right? Then you cannot quite get there because there is energy filling that space in some sense, okay? So that is better shown by the rod. The rod induces in your head this idea that you cannot get there, right? It's not reality. Which one is more real, this or that? Both of those have different aspects of reality, okay? Clearly, this is much, much simpler to look at. By the way, just here, again, one is a strand. The connection here is a sheet. The backbone hydrogen bonds stabilize it. The arrow direction is simply the way the chain runs. Now, simply the way the chain runs, what that means simply, that's the protein is beginning at some point and ending at some point. That's the begin, and that's the end. Okay, so you follow through here on the protein, and you see. Oops, I lost it somewhere. Where's my helix coming out? Somehow I'm getting into a loop. It's this one here. So this is the way the protein runs. Okay, boop. I lost the circle. Somehow this is out. I went back in.
Why do we need the direction?
So why do we need a direction? Why do we need a... So part of this is about recognizing images. So if I plot the arrows differently, then you may immediately see that is something else. seeing something else. You may suspect that may actually be a different protein and may actually do something else. Now, does it matter? Does the directionality matter? There is no reason to explain that it cannot matter, but the observation is it does actually matter. Why does it matter? So it does not matter for folding. It does matter for creating the three-dimensional structure. You cannot turn things around, they will fold differently. Once this three-dimensional object is formed, directionality does not necessarily matter. So once we are on that level of the image, the only thing it matters for is to sort of go and trace the protein. Now, why is that important? That may be important because you get to the beginning. That beginning may contain a different signal, a different part of the sequence, and may do something special. But the core, for the core of it, the directionality actually doesn't matter. You're right. Good. Those are cartoons. Protein cartoons, again, different representation of the same object. There are components of structures. So far, the components, what I talked about so far today, were sort of the smallest components. The smallest components being amino acids, being secondary structure. On the next level, I'm going to get into larger components, so-called domains. Domains are essentially larger subunits. I will also, over the course of the next 4 lectures, get into a little bit why did I present only 2 regular secondary structures. I only presented alpha helix and beta strand. It turns out those are the only ones essentially that we observe. And I will get back to that. But the next topic is a little bit bigger. So the next one is getting into domains. Domains have to do with getting to the level of perceiving, looking at these images and understanding or having some memory of, I have seen that before. And then we get into the problem of how can we compare these three-dimensional objects. One way, of course, is you look at these images and you say, okay, I've seen that. But how can we teach a computer to do that? How can we compare three-dimensional objects and what are the problems there? And how can we make computers realize that 2 things are similar, and realize that 2 things are similar? You immediately, at one point here, the directionality doesn't matter. So what actually is it that makes ultimately 2 protein structures essentially be the same three-dimensional objects, do the same function? Structure comparison, structure domains, next week. Thank you for your attention.